So let's say Bhavya, let's say hypothetically, if you had a, okay, you come to me, I'm a doctor, okay, you're a patient, you look at your hand and you see a little lump in your hand, it's like anomalous. What will you do?
Well, I would wait it out and see if it goes away by itself. If not, then I'll go to.
Yeah, because you don't, I mean, as normal people, we don't immediately assume that it's cancer, right? We don't immediately assume it's cancer. Okay, now what happens is, now that lump is becoming bigger, so that every month it's becoming slightly bigger. Or maybe not every month, it actually grows very slowly. Maybe in a year, it's maybe become slightly bigger. It's not the same. And it's like now reddish a little bit. Now what do you do?
Yes, we'll get it tested.
Yeah, you'll go to the doctor, and the doctor, what do you think the doctor will do? Anyone can answer this question.
Maybe looking at it clinically, if feels there's a need, then get a biopsy or something done.
Yeah, obviously they'll take a biopsy done. They'll first of all surgically remove it, and then take the biopsy and look at the tissue section. And let's say they take a tissue section and they say, okay, it is sort of an early cancer. What do you think the doctor will treat you with?
Maybe like chemical compounds. I mean, like drugs.
Chemical compound. They'll give you some chemical drugs, and let's say it's a regimen that you take 1 tablet a month, 1 tablet a day for 1 month, right? And you think this is not like a Tylenol or paracetamol tablet, right? It's a very bad tablet. It's causing all kinds of side effects, isn't it?
Yes.
Right? And it's sort of debilitating to your healthy function. But you still have to take it because you take it and you take it for a month, and it has been surgically removed, and then it's gone, and now 6 months later, it's gone, but 6 months later you come back, or maybe 1 year later, you come and say, you know what, I'm getting this pain in my chest. What do you think? Like on the right side of my chest. What? And you go to the doctor. What do you think the doctor will do?
ECG.
No, it's not the left side of the chest. The right side of, right side of the chest, not the left side of the chest.
Depends if I'm going to the same doctor or if it's a new person.
Yeah, it's some doctor, some specialist doctor.
Yeah, but somebody who knows the history and knows
somebody who knows the history. Somebody knows the history. It's like, what do you think the doctor will suspect?
A return of cancer.
Return of the cancer, right? And now it's inside. Now you can't surgically remove. Then what will you do?
Will bombard me with maybe
more
chemotherapy, radiotherapy.
Yeah, radiation. Now I'll get radiation. I've done surgery, I've done chemotherapy, and I've done radiation. Okay, now everyone's happy. My radiation looks good. The lung which was infected, what was affected, is now okay. Now things are settled down. I go back to my life. 5 years later, 5 years later now, I get more than frequent headaches. Now, what do you think is happening?
Probably back.
The cancer is spreading, right? So this cycle of cancer going, being treated, going away, coming back, going away, coming back, is remission, relapse, remission, relapse, remission, relapse. And this has been, we know about this from, we all know people in our families and friends who have had cancer, and they go through this process. And whenever you get treatment with these, tremendously bad drugs, they're completely washed out. They lose weight. A lot of the physical strength is gone. Everything is gone, right? So then, okay, they take surgery, they do everything in the brain and everything is done, right? Now, it's now 10 years later after your initial spot that you saw, right? And now it's really bad. Now the tumor has gone to the gut, to the lung, to the brain. You know, it's you see spots in your skin. Now what will you do? You've taken everything. Now what will you do? It's gone. And you've lost all your weight. You've completely degraded. So I'll tell you what we'll do now. I'll tell you now. Till 10 years ago, this patient, people would have said, okay, now let's put this person into a rehab. The last few days, let them live peacefully. Let them go to some meditation and, you know, be with their family, spend their last days in health, right? Until Jim Allison came up with this amazing discovery, not invention, of how the immune system works, gave them one shot of this drug, and the entire cancer went away, and these people are completely cured of their cancer. That is the story of immunology of cancer. That's what's happening today. By harnessing the power of the immune system, you can eliminate the tumor. That's the story I'll tell you. Okay? And how did this come about? So it's a holistic view of cancer. This is me in 1982 using a Macintosh computer in Tata Cancer Institute. And I talk, because I do this presentation many times, I talk a little bit about my experiences. So what I'm going to tell you about today now in the next 30, 40 slides is a 4-decade story of cancer. I'll give you a brief background. I'll tell you a few hallmarks of cancer. I'll tell you what are called the 3 E's of cancer. Clinical trials, challenges, what the next decade is going to look like, and what the radical advances in technology today are even transforming cancer completely. So like we said, the initial tumor starts with a mutation. All tumors start with a mutation. Some mutation has to occur. And why does mutation occur? Mutation occurs, mutations are occurring all the time in our body. You, me, everybody, all the mutations are all with pollution, the food we eat, the random mutations that are occurring. These mutations are constantly occurring in our body, constantly. And when the mutation occurs, something wrong goes with the cell starts trying to divide, right? And then if it's uncontrolled, the cells, they divide into 2 and they become a little lump over here, like you see here in the lung, right? This is what cancer is about. So from 1900 to 2000, almost 100 years, right? And maybe you can even talk about 500 years or even 5,000 years. The only treatment for cancer until the year 2000, which is 25 years ago approximately, was cut, burn, and poison. You know what these are, right? Surgery, radiation, and chemotherapy. That's all we were doing to treat cancer: cut, burn, and poison. Every 10 years or so, there's a journal called Cell. So those of you who probably read this journal, Cell, know that Cell is a journal which is the highest impact factor ever. Like if you have a paper in Cell, it's huge. It's very, very difficult to get a paper published in Cell. It is the landmark paper of science. That's how big the journal is. And the journal, its editorial board, every 10 years asks the leading exponents of those field to write what is going to happen in the next 10 years. So Hahnemann and Weinberg in the year 2000 were asked to write a review article to predict what is going to happen from 2000 to 2010. And what they wrote in this manuscript was basically that we will have with the advances in technology from 2000 to 2010, we will have a better understanding of how cancer cells replicate. And instead of better chemotherapies will come, we'll be targeting cells and how how to kill them properly. We can block angiogenesis by stopping blood vessels to grow inside the tumor cells. And we'll be understanding all of these biologies much better from 2000 to 2010. And that's exactly what happened. And from 2000 to 2010, fantastic drugs were discovered. In 2000, human genome was sequenced, so we knew a little bit more about how the genetics of cancer also was working. And these amazing drugs came into the market, which are called targeted gene therapy. So instead of giving chemotherapy, which basically kills every growing cells in your body, there were drugs which were specifically killing only the cancer cell. So that was revolutionary in of itself. But they were not cures. They were not cures. Another fantastic thing happened in this 2000 to 2010 period. There's a person in the middle of this slide. His name is Robert Schreiber. He's a professor at University of Washington in St. Louis. He went to undergrad. When I teach this in colleges and schools, I tell the history of these people also because it's inspirational to see where they came from. He comes from Buffalo, New York, which is like a small place, but has gone on to do some fantastic work. So what he's getting the award for from two stalwarts of the industry. This on his right is Jim Allison, who's the Nobel laureate, whose story I'm going to tell you. And on his left is Laurie Glimcher, who's the head of the Dana-Farber Institute, which is the Cancer Institute of Harvard University. So they're giving him this award for what? For describing how cancer is caused and the phenomenon called immunoediting. Looks like a busy slide, but I'll break it down a little bit and I'll tell you the story behind it. So when I told you, Bhavya, that you had a lump in your hand in the hypothetical situation, what was happening is we get lumps in our hands all the time and we don't even know. What's happening then? Either carcinogens, radiation, viral infections, inflammation, all kinds of inherited genetic mutations are constantly occurring in your body, right? But our immune system is healthy. It is quickly able to recognize these mutations and kill the cells which are mutant. And this first E of the three E's is called elimination. So this elimination of mutant cells is always occurring in our body. It's not like our bodies are not undergoing mutation. They're undergoing mutation, but our healthy immune system recognizes it and kills these tumor cells all the time. This is happening every day in all our lives. We don't even know, right? In the battle between cancer and the immune system, what happens is one day that mutation will occur, and for some reason, maybe you got a bad infection, or you got some other infection. Let's say you got flu, or you got COVID, or you got some other infection where somehow your immune system slightly weakened on a week or so, or 2 weeks. Overall immune system went down. When your immune system went down, that one mutated tumor cell quietly becomes 2 cells, just very quietly, unbeknownst to the immune system. Now the immune system is busy trying to solve another problem. This mutant problem is not solved by the immune system. And this phase is called equilibrium phase, where there are tumor cells in your body, your immune system is healthy, nothing goes wrong, and these tumor cells are so few that they don't have any clinical symptoms. This equilibrium phase can last for 10 years, maybe 20 years, maybe 40 years. The tumor cells are very resilient. They will stay quietly as long as they need to, right? So in the example that I gave you, Bhavya, so see a lump in your hand, right? And there's a significant lump. The tumor has been there for already 20 years. After 20 years, you'll see the lump. So when women see, when you're feeling for breast cancer and you're feeling around and anyone feels like you have an abnormal kind of situation, by the time you see it, it's already been 20 years that the cancer has been in your body. That's the And what happens after 20 years? The immune system now finally gives up, and the cancer cells now take off. And when you see the escape, what I've shown you as escape is when, Bhavya, you see the first lump. Escape is not when you've got all the metastasis and your brain is affected. That's not escape. The first lump that you see, that is the escape. This phenomenon was experimentally demonstrated by Robert Schreiber, and he calls it the immunoediting process. And I won't go into the details right now about the various ways the immune system escapes, or the cancer cell escapes the immune system, but some of them are listed here. So there is a very detailed cellular basis of this immunoediting. You know, we now know a lot of cells that are involved in this in maintaining elimination, in inducing equilibrium, and the role of various kinds of cells that are involved in the escape of the immune system. So we'll study all of these in the course of the immune system. Okay, now what happened is, remember that journal I told you, Cell? It is now 2011, and it's now time to write about what's going to happen in 2010 to 2020. And so Hahnemann and Weinberg, these are again the same two authors, wrote another article saying now what's going to happen in the next 10 years is we are going to understand how the immune system works against cancer in the next 10 years, and we're going to have immune-mediated drugs that are going to come into the market. And that's exactly what happened. There was a drug called Yervoy, which is made by Bristol Myers Squibb, which is called an anti-CTLA-4 antibody or CTLA-4 drug. And you can see here breast cancer on the left side. This woman has a breast cancer, a huge breast cancer here. After treatment of one dose with breast cancer with this drug, that breast cancer is gone. Gone. Woman is cured. So what is this drug? What is this drug? To understand this drug, I have to tell you about basic immunology. What happens is, and we had started talking about antigen presentation, MHC class 1 and class 2. We had talked about it last time, right? So what happens is, and this is sort of this phenomenon of immune responses were discovered in 1990s. This whole thing that I'm telling you is only about 20, 30 years old. So antigen, in this case, a cancer antigen, is taken up by an antigen presenting cell. It is processed and broken up into small peptides and presented on the surface of MHC class 2. This cancer antigen is recognized by T cells, CD4 positive T cells, by its receptor called the T cell receptor. And this T cell receptor, when it recognizes that antigen, it delivers a signal into the T cell. Signal is a biochemical activation signal. It activates the signal. This signal upregulates or induces expression of another molecule, for example, CD40 ligand, which will bind to CD40 and then activate another cell. This activation, it's a cascade event. It'll activate another signal called CD8, and this will activate a signal called signal 2, right? Think of this as the analogy for this is like a nuclear bomb, right? So a country has a nuclear bomb. First of all, the prime minister or the president of the country has to give authorization to set off the nuclear bomb. Somebody has to take accountability for that, right? And usually when you have a nuclear bomb, you have two, like a movie, right? You need two keys. One person has one key and the other person has another key. Only then will it be set off. That's what the T cell also requires. It requires two sets of keys, one from the T cell receptor, signal 1, and another independent signal called CD28, which is called signal 2. And a T cell is like a nuclear bomb for our body. If you just activate T cells with one signal, it'll go and kill your entire immune system, entire body. That's what happens in autoimmune disease, right? But in this case, in this normal situation, you need two signals. You actually need more than two signals. You need, in this case, the third signal. But for our discussion, this third signal is not critical. These two signals, then, when activated, unleash the activity of T cells, and these T cells will now go and hunt down the tumor and kill it. All right? This is happening every day in our lives. This is the elimination mechanism. I've oversimplified the presentation by telling you that there is T cell receptor and CD28, but there are many other signals along the way. Now, before I go there, I want to tell you that in the 1990 to 2000 or 2010 timeframe, T cell receptor was recognized, antigen presentation was recognized, CD28 binding to B7-2 was being all not recognized, being discovered. These genes were being discovered. They were not known. Somebody found ICOS, somebody found PD1, somebody found FAS, somebody discovered CD40. All of these exciting discoveries were happening, right? So now people started saying, if I give signal 1, if the T cell gives signal 1 through T cell receptor and signal 2 through CD28, maybe a third signal through CTLA4, which is a third signal, will make the immune cell even stronger. It makes sense, right? If I have 2 signals, it'll be strong. If I give 3 signals, it'll be even stronger. Isn't it? That makes logical sense. So everybody and their brother, by the way, including me, I was also doing the experiments at this time and with this kind of system. We used anti-CTLA4 antibody, etc., and tried to stimulate these T cells. And to our surprise, if in a culture plate, if you take a T cell and you stimulate it with T cell receptor, and then you stimulate it with CD28, and you stimulate it with anti-CTLA4, you will get massive activation. So let's say you get a signal if you're measuring some output, let's say whatever that output is, it has a unit of 1. If T cell receptor, that signal 1 induces the output of 1, signal 1 plus 2, which is T cell receptor plus CD28, will give you an output of 10. Okay, so it induces a very strong signal. If you now put anti-CTLA4, the signal becomes 10,000. So if you give anti-CTLA4 antibody, or if you activate that third signal, you get 10,000 units of that signal. You activate the T cell enormously if you allow CTLA4 B7 interaction to occur. So everybody thought, oh wow, I can easily stimulate the immune system and get rid of cancer. What a great idea. I'll give signal 3 and activate the immune system, and then I can get the Nobel Prize. Everybody was trying it, everybody failed. Jim Allison did a very cute experiment, very simple experiment. And this is, I think, what I love about science is that the simpler the experiment, the more real it is, the more real to the truth it is. If you try to make a convoluted experiment, it's probably not right. So what he did is, he took a simple experiment. He took a mouse, and he induced a cancer. He induced a cancer in the mouse. And then he treated it with the control, which is nothing, basically PBS. And then over time, as you can see, the control mouse grows. The tumor in the control mouse grows as expected. Nothing surprising here. In 1990s, when we were studying this signal 1 and signal 2, we would have assumed that if you give anti-CTLA4 or if you give anti-CD28 antibody, which induces signal 2, signal 1 plus signal 2, you should activate the T cell, and the T cell should kill the tumor, isn't it? That's what we would have thought. But surprisingly, if you give anti-CD28, only give the signal 2, it doesn't kill the tumor. It actually makes it worse. You can see the curve. The tumors are growing faster if you give signal 2. What the hell's happening? Then he gave CTLA4. And there's a beautiful story. There's a book about him. There's a movie about this story called Breakthrough. I recommend seeing it if you're interested. What he did is, what his postdoc did was, at that time, he had these mice and he treated these mice. And I think it was the Christmas holidays. So he treated this mice and he said, nothing's working anyway. Let me just go on a holiday and come back. He treated the mice with anti-CTLA and came back from the holiday. And the mice were the tumor was completely gone. All the control mice had died, but the animals that were treated with CTLA4 were absolutely, as if they've never been given tumor, completely gone. So this was the result of that experiment. What happened? So what had happened was, as usual, the T cells were activated. They got signal 1, they got signal 2. Then what happened is, then they got activated. Then what happens is CTLA4, which is a receptor on the T cell, which comes up a little bit later. It is not present in the earlier phases of the T cell activation. It comes up a little bit later. And like I told you previously, all of us in the world were thinking CTLA4 delivers signal 3, which it does, right? And we thought that signal 3 is this huge activator of T cells. It is not. It is actually, it shuts the immune system down. It's not an activator of the immune system. It's actually an inhibitor of the immune system. And that's what he got the Nobel Prize for making this discovery. Because everybody else was thinking that it was an activator, but it's an inhibitor. So how does an inhibitor cure cancer? So how did this work to cure cancer? What happened was, in normal situation, in that elimination phase of the cancer, what happens, you get signal 1 and signal 2. This is happening every day in our lives. T cells get activated and they kill the tumor, right? Now, for this story, I have to tell you, like CTLA4, there's another molecule called PD1, but it does exactly the same thing as CTLA4. What happens is the tumor starts getting very smart. What the tumor does, it upregulates a molecule called PDL1. It didn't have it before, but it now has a new molecule on its cell surface called PD1. Now, when the T cell comes and tries to attack it with signal 1 and signal 2, what happens is, since T cells express PD1, the PDL1 on the tumor binds to PD1, and this tumor shuts the immune system down. So the T cell can't kill the tumor anymore. And so what Jim Allison did is he made an antibody to PDL1, blocked that interaction between PD1 and PDL1, and now the tumor cell can be killed. So he inhibited the inhibitory signal, which was preventing the T cells from killing the tumor. Inhibiting the inhibitory signal. That's what he got the Nobel Prize for, and that's what happens when you treat with anti-PD1 or CD1 antibody. And so that's it. That's the story I want to tell you about cancer. And now cancer is this therapy is being used to treat cancer, enormously. So that's the essential story of cancer. Now cancer therapy is being used every day. Let me see if there are questions. Did you understand? Was it too complex? There was still some immunological concepts in it, but
You go back to the slides. Believe so. There's a reversible arrow between elimination and equilibrium, right? But equilibrium,
yes, yeah.
Is not.
Yeah, 1 second. Let's keep. I have to get out of presentation mode. Yeah. What are you saying?
I was asking if I saw it correctly.
I'll share.
The elimination and the equilibrium state are reversible, right? So there's a
reversible. Yeah, it's going back and forth and back and forth. Correct.
But equilibrium to escape is not reversible.
It's not reversible. Generally not reversible unless you come up with some therapy in the middle. Correct. Yeah. So this in the story, this is a unidirectional thing. This is happening every day in your body.
Mm.
Then this actually, from equilibrium, this is a very dynamic state. This is not happening. Some days you're good. Some days the cells are growing. So this is a war that is going on all the time. Yeah. So I'll show you
also,
one last. Yeah, go ahead.
Yeah, before that, also in your example, hypothetical situation, me having the lump of time. I mean, the times that you mentioned that the relapse occurs was kind of logarithmic or quadratic, like that. Is it?
Yeah, it is like that. Most cancers are not as aggressive. They grow very slowly. There's a theory. I mean, nobody is going to ever test this hypothesis but in this, based on this immunoediting theory, if you don't treat a cancer patient with anything, you might actually live longer. And because you try to do you do chemotherapy and radiation therapy and this therapy and that therapy, when you treat the cells, the tumor cells that evolve out of that, the remaining cells that were resistant to those tumors are the ones that are going to come out, right? Are going to survive. And so by treating, you're making the tumor more aggressive.
So it sounds more like antimicrobial resistance, right? If okay.
Correct. I mean, there are a lot of more. We're going to go through. Yeah, go ahead.
So I have one more question regarding that only. So you mentioned about mutation and then cancer. So does it mean that any cell which is mutated can become a cancerous cell in 10 years, or there is some criteria which it has to go through?
Yeah. I mean, a genetic mutation is necessary, but not sufficient to make it a cancer.
Okay.
There are many other factors. We all might have mutations, but our body sort of controls that mutated cell and doesn't allow it to grow.
Yes, that makes sense. And I have one. Yeah, go ahead, Bhavya.
You can go ahead.
Okay, thank you. So in the CD28 signal activation, you said that that's signal 2, but also you mentioned that signal 1 is somehow affecting signal 2, right?
Yeah.
So,
signal 1 induces signal 2. Correct.
Signal 1 induces signal 2. So in this case, actually, it seems that there's only one signal, right? Because it is inducing signal 2. So like the example
logically,
of cookies.
Logically, it's only one signal, but it's sort of a convoluted way of saying that, but two separate receptors are delivering.
Separate receptors are there. Okay, got it.
No, that's a good thought. Okay, so I'm going to come back to antigen presentation after this. Go ahead, keep asking questions.
Oh. So in the experiment, right? So CD28 and the control.
Yeah.
Control is not growing at the rate. I'm not sure about the time scale, but compared to both the treatments, control initially, the tumor size wasn't growing, right? So
well, in that experiment, the control, the tumor cell grows regularly. I mean, it was on that 45-degree angle, right? So over time, it's growing.
Okay.
And then when you treat with anti-CTLA-4, the curve is flat because once the tumor, it doesn't grow, because the y-axis is tumor size.
Yeah. Got it.
So let me make this comment, and then I'll go back to the antigen presentation to put it all back in context of why we are studying what we are studying. Is I told you the history of oncology has been, for 100 years, it was cut, burn, and poison. In 2000, in those 10 years, targeted therapy started becoming better, where you started getting very specific drugs, with less side effects, not zero side effects. Then 2010, immunotherapy came about, and today, immunotherapy is great. Now it's 2022 now, or 2020. The next 10 years, paper has come out in Cell. 2020. What's going to happen in the next 10 years? And what they've predicted is something absolutely fantastic. And this is written by a different group of people. They are not the same authors. What they have written is in the area of cancer, and not just in cancer, in many diseases. Over the last 5,000 years of medicine, humankind has been, and doctors especially, and physicians and scientists and all of us have been focused on the mechanism of disease, how disease is caused. Very few people, in fact, very, very few people are studying why are we healthy. So this paper is about the next 10 years is going to be a better understanding of why people are healthy. I mean, the irony of this for me is that, I say this is that we say we go to healthcare centers. We call hospitals as healthcare centers. They're not healthcare centers. They're disease care centers. The healthcare center is your yoga studio, your gym, your park, your trek. These are healthcare centers. So pay attention to not getting sick rather than trying to figure out why you're getting sick. Of course, understanding diseases is allowed to understand biology in general, but that's what this paper is about. And if you're interested in these articles and you want to read, they're very, very scientific, but you can read as much as you can get. I'll put all of these papers into our link, and you can read all these hallmark papers. They're just fun papers to read. It's like reading that what is that, Orwell's 1984 book. He predicted what was going to happen in 1984, right? So yeah, Jaya.
Yeah, I just wanted to. I've been reading something about the salutogenesis movement of looking at why and what has kept people healthy, and it started with a study of survivors of the Nazi camps, and then there's a lot of work I understand that's informing the public health, and so I was like that always is of interest to me, even in my work. How can systems be healthy, communities be healthy, rather than only be looking at problem fixing? So yeah, I got
Exactly.
reminded of that when you were saying that.
Yeah, no, absolutely. So in the last 10 minutes or so, for today, let me just quickly revise, and we can go through this in more detail again, but let's go through that antigen presentation talk that I told you about, right? So think in the context of the cancer cell that we were talking about. So imagine this is the cancer. It is actually tumor cells. What is tumor antigen? Tumor antigen, or the tumor cell, is an intracellular antigen, meaning it is the tumor inside the cell, right? The mutation has occurred, and the mutation that has occurred is present inside the cell, right? And so when the tumor cell, let's say this is the mutated tumor antigen, the mutated tumor antigen enters the proteasome, and it is cleaved into small peptides, and these peptides are loaded in the endoplasmic reticulum on the MHC class 1 molecule, and this is the tumor antigen that is being displayed to the T cell, right? And this tumor cell antigen that is being displayed is going to be delivering what we called this T cell interaction is delivering what we call signal 1. Remember, we called signal 1 in the cancer story. This T cell receptor is induced, this peptide is inducing signal 1 and activation of the T cell. Signal 1 is not sufficient to activate the T cell. You need signal 2. If the strength of the signal is not strong enough, you will never get signal 2.
If I may interrupt,
yeah,
my understanding. So signal 1, like, isn't it differentiating between self and non-self? So in the previous last week, so but then if it's cancer cell, isn't it self?
No, it's non-self, right? Because when a mutation occurs, that what was self became non-self. A mutation will make it non-self.
Got it.
Right? Unlike a virus or bacteria, the origin of the cancer cell is always self, the original protein. It has some mutation and then it becomes non-self. So what happens is cancer cells are not very immunogenic. They're not able to induce a strong immune response because majority of the cancer protein is self. Only that one small bit is non-self, right? So it is not a strong activator of the immune system. That is another problem in treatment of patients with cancer, activating immune systems in cancer, because it is not a strong antigen. Anyway, let me pause here. And this is I just wanted to, and for the MHC class 2, even though we call the antigen and the tumor cell as intracellular antigen, when the tumor cell is there, it's outside. The tumor cell can be engulfed by, let's say, a macrophage, and when it's engulfed, the tumor antigens are then loaded onto MHC class 2, which then activates CD4 positive cells, and that is delivering signal 1 to CD4 positive cells. Yeah. So that's where we ended last time. Is it clear? Because we have to also move on, right? So I'm going to pause here for today. Next time we'll look at how instead of going into this complex CD1 part right now, what we'll do is next week we'll talk about how the antigen is recognized by the T cell. We'll talk about how the T cell receptor and the antibody receptor is made in the body, and how does it recognize this presented antigen. That we'll talk about next time.